हेलो लर्नर्स, वेलकम बैक टू आवर चैनल बी क्यूरियस। आज हम पढ़ने जा रहे हैं बायोमोलिक्यूल्स। इसमें हम कवर करेंगे हाउ टू एनालाइज केमिकल कंपोजिशन, प्राइमरी एंड सेकेंडरी मेटाबोलाइट्स, बायोमैक्रोमोलिक्यूल्स, प्रोटीन्स, पॉलीसेकेराइड्स, न्यूक्लिक एसिड, स्ट्रक्चर ऑफ प्रोटीन्स, एंजाइम्स. So there is a wide diversity in living organism in our biosphere. What are the difference between two lists? In absolute terms, no such differences could be made out. All the elements present in sample of earth crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is high. How to analyze chemical composition? We can take any living tissue like a vegetable or piece of liver and grind it in trichloroacidic acid CI3CCOOH using a mortar and pestle. We obtain a thick slurry. If we were to strain this through a cheesecloth or cotton, we would obtain two fractions. One is called the filtrate and other technically the acid solution pool, and the second the retentant or the acid insoluble fraction. All the carbon compound that we get from living tissue can be called biomolecules. However, living organisms have also got inorganic elements and components in them. How do we know this? A slight difference but destructive experiment has to be done. One way, a small amount of living tissue and dry it, all the water evaporates, the remaining material gives a dry weight. Now if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form (CO2 and water vapor) and removed. What is remaining is called ash. This ash contains inorganic elements like calcium, magnesium. Inorganic compounds like sulfate, phosphate are also seen in the acid solution fraction. Therefore, elemental analysis gave elemental composition of living tissue in the form of hydrogen, oxygen, chlorine, carbon. From a chemistry point of view, one can identify functional groups like aldehydes, ketone, aromatic compounds, but from a biological point of view, we shall classify them into amino acids, nucleotide base, fatty acids. Amino acids are organic compound containing an amino group, and an acidic group are as substituents on the same carbon, that is the alpha carbon. Hence they are called alpha amino acid. They are substituted methane. There are four substituents groups occupying the four vacancy positions. These are hydrogen carboxyl group, amino group, and variable group designated as R group. Based on the nature of R group, there are many amino acids. However, those which ones occur in protein are only of 20 types. The R group in these proteinaceous amino acid could be hydrogen. The amino acid is called glycine. Methyl group alanine, hydroxyl methyl serine. Three of the 20 are shown in figure 9.1. So these are the pictures. Here, first picture showing sugar, carbohydrate, which is glucose and ribose. Second is amino acid, which is showing glycoside, alanine and serine. And third one is fat and oils, which is palmitic acid, glycerol and triacid. triglyceride, and R3 are fatty acids, and phospholipid, lecithin, and fourth is nitrogen base, which is alanine and purine, uracil and pyrimidine. Fifth is nucleosides, which is adenine and uridine, and last one is nucleotide, which is adenyl acid. The The chemicals and physical properties of amino acid are essential of amino, carboxyl, and the R functional groups. Based on number of amino and carboxyl group, there are acid, glutamic acid, basic, lysine, and neutral is valine amino acids. Similarly, there are aromatic amino acids like tyrosine, phenylalanine, tryptophan. A particular property of amino acid is the ionization nature of NH2 and COOH group. Hence, in solution of different pH, the structure of amino acids changes. This is called Zwitterion form. Lipids are generally water soluble. Okay, lipids are water soluble. They could be simple fatty acid. They could be simple fatty acids. A fatty acid has a carbonyl group, carboxyl group attached to a R group. Attached to R group. The R group could be a methyl CH3, ethyl C2H5, higher number CH2 group. For example, palmitic acid has 16 carbons including carboxyl carbon. Aryl arachidonic acid has 20 carbon atoms including the carboxyl carbons. Fatty acid could be saturated or unsaturated. Saturated or unsaturated. So saturated having without double bond, and unsaturated having one or more C double bond C bonds. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Here the fatty acids are found esterified with glycerol. They can be then monoglyceride and diglyceride and triglyceride. These are also called fats, oils, based on melting points. Oils have lower melting point, hence remain as oil in winter. Can can you identify a fat from the market? Some lipids have phosphorus and phosphorylated organic compound in them. These are phospholipids. They are found in cell membrane. So this is found in cell membrane. Lecithin is one example. So phospholipid found in cell membrane and lecithin is the example. Some tissue, especially the neutral tissue, have lipids with more complex structure. Living organisms have a number of carbon compound. So living organisms have a number of carbon compound in which heterocyclic rings can be found. Some of these are nitrogen bases: adenine, guanine, cytosine, uracil, thymine. So these are the nitrogen base. When found attached to sugar, they are called nucleoside. Okay, so very clear it is. Adenine, guanine, cytosine, and uracil, thymine all are attached with sugar. So these are called nucleosides. If a phosphate group is also found esterified to sugar, they are called nucleotide. Phosphate group also found esterified to the sugar, so they are called a nucleotide. Nucleotide: adenosine, guanosine, thymidine, uridine, cytidine are nucleosides. Adenyl acid, thymidyllic acid, guanylic acid, uridylic acid, cystidylic acid are nucleotides. Nucleic acid like DNA and RNA consist of nucleotide only. DNA and RNA function as genetic material. Primary and secondary metabolites. So, what are primary and secondary metabolites? So, if we see these table 9.3, all are the secondary nucleotides. The most exciting aspect of chemistry deal with isolating thousands of compounds, small and big, from living organisms, determining their structure and, if possible, synthesis them. If one were to make list of biomolecules, such a list would have thousands of organic compounds, including amino acids, sugars. For reasons that are or in animal tissue, one notice the one notice the presence of all such category of compounds shown in 9.1. So these are the compounds, biomolecule compounds. These are primary metabolites. However, when one analyzes plant, fungus, and microbial cells, one would see thousands of compounds other than these primary metabolites: alkaloids, flavonoids, rubber, essential oil, antibiotics, color pigments, scents, gums, spices. These are called secondary metabolites. While primary metabolites have identifiable functions and play roles in normal physiological processes, we do not at the moment understand the role or function of all the secondary metabolites in host organism. However, many of them are useful to human welfare: rubber, drugs, spices, scents, pigments. Some secondary metabolites have ecological importance. In the later chapter, biomacromolecules. There is a one feature common to all those compounds found in acid soluble pool. They have a molecular weight ranging from 18 to around 800 Dalton approximately. The acid insoluble fraction. The acid insoluble fraction has only four type of organic compound. Four type of organic compound: proteins, nucleic acids, polysaccharides, lipids. These classes of compound, with the exception of lipids, have molecular weight in the range of 10,000 Dalton and above. For this very reason, biomolecules, chemical compound found in living organism, are two types. One, those who, one, those which have molecular weight less than 1000 Dalton and are usually referred to as micromolecules of simply biomolecules, while those which are found in the acid insoluble fraction are called macromolecules or biomacromolecules. The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Then why do lipids, those molecules weighted do not exceed 800 Dalton, come under acid insoluble fraction, micromolecular fraction? Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structures like a cell membrane and other membrane. When we grade a tissue, we are disrupting the cell structure. The cell membrane and other membrane are broken into pieces and form vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicles get separated along with and the acid insoluble pool, and hence in the macromolecules fraction. Lipids are not strict, strictly macromolecules, not strictly macromolecules. The acid soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid-insoluble fraction. Together, they represent the entire chemical composition of living tissue or organism. Proteins. Proteins are polypeptides. They are linear chain of amino acids linked by peptide bonds. Each protein is polymer of amino acids. Each protein is a polymer of amino acid as there are 20 types of amino acids: alanine, cytosine, proline, tryptophan, lysine. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n number of times. This information about the amino acid content is important as later in your nutrition lessons. You will learn that certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non essential. The latter are those which our body can make, while we get essential amino acid through our diet food. Proteins carry out many functions in living organisms. Proteins carry out many functions in living organisms. Some transport nutrients across cell membrane, some fight infectious organisms, some are hormones, some are enzymes. Collagen is the most abundant protein. So collagen is the most abundant protein in animal world and ribulose biphosphate, ribulose biphosphate, carboxylase oxygenase Rubisco is the most abundant protein in the whole of the biosphere. Polysaccharides. The acid insoluble pellet also has polysaccharides, carbohydrates, as another class of macromolecules. Polysaccharides are long chains of sugar, long chains of sugar. There are threads, likely a cotton thread, containing different monosaccharides as a building blocks. For example, cellulose. Cellulose is a polymeric polysaccharide consisting of one type of monosaccharides, glucose, that is glucose. Cellulose is a homopolymer. Cellulose is the homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissue. Animals have another variant called glycogen. Insulin is a polymer of fructose. Insulin is the polymer of fructose. In a polysaccharide chain, say glycogen, the right end is called the reducing end. Right hand is called reducing hand and the left hand is called the non-reducing end. It has branches as shown in the form of cartoon. Starch forms helical secondary structure. In fact, starch can hold I2 molecules in the helical portion. The starch I2 is blue in color. Cellulose does not contain complex helicals and hence called hold I2. Plant cells Plant cell wall are made of cellulose. Paper made from plant pulp and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have a building block, amino sugars and chemically modified sugar, glucosamine and acetyl glucosamine. Exoskeletons of arthropods, for example, have a complex polysaccharide called chitin. These complex polysaccharides is mostly homopolymers. Nucleic acids. The other type of macromolecules that one would find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides. Together with polysaccharides and polypeptides, these comprise the true macromolecular fraction of any living tissue or cell. The nucleic acids, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is heterocyclic compounds, second is monosaccharides, and the third is phosphorylic acid phosphate. The heterocyclic compounds the heterocyclic compounds in nucleic acids, nitrogenous bases, adenine, guanine, uracil, cytosine, thymine. So, in heterocyclic compound, nucleic acid are a nitrogenous base: adenine, guanine, uracil, cytosine, thymine. Adenine and guanine are substituted purines. Purines. Adenine and guanine are the substituted purines, while the rest are substituted pyrimidine. The skeleton heterocyclic ring. The skeleton heterocyclic ring is called purine and pyrimidine, respectively. The sugar found in polynucleotide is either ribose, a monosaccharidic peptose, or 2-deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid (DNA), while that which contains ribose is called ribonucleic acid (RNA). Structure of proteins. Proteins, as mentioned earlier, are heteropolymers containing strings of amino acids. Structure of molecules means different things in different contexts. In organic chemistry, the structure invariably refer to molecular formula: sodium chloride, magnesium chloride. Organic chemistry always write a two-dimensional view of molecules while representing the structure of the molecule. Benzene, naphthalene. Physicists conjure up the three-dimensional views of molecular structure, while biologists describe the protein structure at four level. The sequence of amino acids, the positional information is a protein in a protein, which is the first amino acid, which is second, and so on, is called primary structure. A protein Primary structure of protein. A protein is imagined as a line, or the left end represented by the first amino acid, and the right end represented by the last amino acids. The first amino acid is also called N-terminal amino acids. The last amino acid is called the C-terminal amino acid. A protein thread does not exist throughout as the extended rigid rod. The thread is folded in the form of helix, similar to the revolving staircase. Of course, only some portion of the protein thread are arranged in the form of helix. In proteins, only right-handed helicals are observed. Other regions of the protein thread are folded into other forms in what is called secondary structure. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to a tertiary structure. So this is the primary structure, which is like a simple thread. Secondary structure, which is helical, alpha helix are formed, beta plated sheet, form secondary structure, and the hydrogen bond disulfide bond containing are a tertiary structure. This gives us a three-dimensional view of a protein. Tertiary structure is absolutely necessary for the many biological activity of proteins. Some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other, linear string of spheres arranged one upon each other in the form of cube or plate, the is the architecture of a protein, otherwise called quaternary structure of protein. So this structure is quaternary structure of protein. Adult human globulin adult human hemoglobin consists of four subunits. Two of these are identical to each other, hence two subunits of alpha type and two subunits of beta type together constitute the human hemoglobin. Enzymes. Almost all enzymes are proteins. There are some nucleic acids that behave like enzymes. These are called ribozymes. One can depict an enzyme by a line diagram. An enzyme, like any protein, has a primary structure, amino acid sequence of the protein. An enzyme, like any protein, have has the secondary and tertiary structure. When you look at the primary structure, when you look at a tertiary structure up to itself, the chain cis-cross itself, and hence many cervix and pocket are made. One such pocket is the active site. An active site of an enzyme is cervix or pocket into which the substrate fits. Thus, enzymes through their active site catalyst reaction at a high rate. Enzyme catalysts differ from inorganic catalysts in many ways, but one major difference needs mention. One major difference needs mention. Inorganic catalysts work efficiently at high temperature and high pressure, while enzymes get damaged at high temperatures, say about 40 degrees Celsius. However, enzyme is isolated from organisms who normally live under extremely high temperatures. Hot vents and sulfur strings are stable and retain their catalytic power even at high temperature up to 80 degrees to 90 degrees Celsius. Thermal stability is thus an important quality of such enzymes isolated from the thermophilic organisms. Chemical reactions. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compounds undergo two type of changes. A physical change, simply referred to a change in shape without breaking of bonds. This is a physical process. Another physical process is a change in state of matter, while ice melt into water or when water becomes a vapor. These are also physical processes. However, when bonds are broken and new bonds are formed during transformation, This will be called a chemical reaction. For example, BaOH2 plus H2SO4 give rise to BaSO4 plus 2H2O. In an inorganic chemical reaction, similarly, hydrolysis, breakdown of water, hydrolysis of starch into glucose is an organic chemical reaction. Rate, a physical or chemical process, refer to the amount of product formed per unit time. It can be expressed as rate delta P upon delta T. Rate can also be called velocity if the direction is specified. Rates of physical and chemical processes are influenced by temperature, among other factors. A general rule of thumb is that rate doubles or decreases by half for every 10 degrees Celsius change in either direction. Catalysis, catalysts, catalyzed reactions proceed at rates vastly higher than that of uncatalyzed ones. When enzyme-catalyzed reactions are observed, the rate would be vastly higher than the same but uncatalyzed reaction. For example, carbon dioxide plus water give rise to carbonic anhydride. In presence of carbonic anhydride, give carbonic acid H2CO3, and the direction is and the reaction is reversible. It carbonic acid reverse to give water. In the absence of any enzyme, this reaction is very slow. In the absence of any enzyme, this reaction is very slow, with about 200 molecules of H2CO3 being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically, with about 6 lakh molecules being formed every second. The enzyme has accelerated the reaction rate by about 10 million times. The power of enzymes is incredible indeed. There are thousands of types of enzymes, such each catalyzing a unique chemical and metabolic reactions. A multi-step chemical reaction, when each of the step is catalyzed by the same enzyme complex or different enzymes, is called a metabolic pathway. For example, glucose give 2 pyruvic acid is actually a metabolic pathway in which glucose becomes pyruvic acid through a 10 different enzyme catalyzed metabolic reactions. When you study respiration, you can relate glucose to 2 pyruvic acid cycle. You will study these reactions in chapter 12. At this stage, you should know that this very metabolic pathway, with one or two additional reactions, give rise to variety of metabolic end products. In a skeletal muscle, under anaerobic conditions, lactic acid is formed. Under normal aerobic conditions, pyruvic acid is formed. In yeast, during fermentation, the same pathogen Same pathway leads to production of ethanol alcohol. Hence, in different conditions, different products are possible. How do enzymes bring about such high rate of chemical conversions? To understand this, we should study enzyme a little more. We have already understood the idea of an active site. The chemical or metabolic conversion refer to a reaction. The chemical which is converted into a product is called substrate. Hence, enzyme protein with three-dimensional structures, including an active site, convert a substrate S into a product P. Symbolically, they can be depicted as S gives product P. It is now understood that the substrate S has a bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of an ES complex. E stand for enzyme. E stand for enzyme. This complex formation is a conscient phenomenon during the state where substrate is bound to the enzyme active site. A new structure of the substrate called transition state structure is formed. Very soon after the expected bond breaking making is completed, the product is released from the active site. In other words, the structure of substrate get transformed into the structure of product. The pathway of this transformation must go through the so-called transition state structure. Transition state structure. There could be many more alternated structure states between the stable substrate and the product. Implicit in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to energy status of the molecule or the structure. Hence, when we look at the pictorially through a graph, it looks like something in figure 9.4. So, concept of activation energy. X-axis show progression of reaction and y-axis show potential energy. In this structure, two states are given. First is substrate S and second is transition state. Activation energy without enzyme. It's it's a high in peak activation energy without enzyme and activation energy without enzyme. Sorry, activation energy with enzyme. Two states are giving. First is activation energy without enzyme and second is with enzyme. And the substrate showing constant constant state and the product showing progression of reaction. Y-axis represent the potential energy constant and the x-axis represent the progression of the structural transformation or state through the transition state. You would notice two things. The energy level different between S and P. Substrate and product having different energy levels. If P is at a low level then S, the reaction is an exothermic reaction. Exothermic reaction. One need not supply energy in order to form the product. However, whether it is an exothermic or spontaneous reaction. Exothermic and spontaneous reaction, or an endothermic reaction. Requiring reaction, the S has to go through a much high energy state or a transition state. The difference in average energy content of S from that of this transition state is called activation energy. Activation energy. Enzyme eventually bring down this energy barrier, making the transition of S to P easily, more easily. Nature of enzyme action. Each enzyme each enzyme has a substrate binding site in its molecule so that a high reactive enzyme substrate complex is produced. This complex is shorter lived and dissociates into its products and the unchanged enzyme with an intermediate formation of enzyme product complex. E plus S enzyme plus substrate. ES gives enzyme product EP. E plus P enzyme plus P. So the catalytic cycle of an enzyme reaction can be described in the following steps. First, the substrate bind to the active site of the enzyme, fitting into the active site. The binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. The active site of the enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate and the new enzyme product complex is formed. The enzyme releases the products of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. Factors affecting enzyme activity. So there are factors which affecting enzyme activity. Activity of an enzyme can be affected by a change in the conditions which can alter the tertiary structure of the protein. These include temperature, pH, change in the substrate concentration or binding of specific chemicals that regulate its activity. Temperature and pH. Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at particular temperature and pH called the optimum temperature and optimum pH. Activity declines both below and above the optimal value. Low temperature preserves the enzyme in a temporary inactive state, whereas high temperature destroys enzymatic activity because proteins are denatured by heat. Concentration of substrate. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity Vmax, which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzyme molecules to bind with the additional substrate molecules. The activity of enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of the chemical shuts off shut off enzyme activity. The process is called inhibition and the chemical is called inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibit the activity of the enzyme. It is known as competitive inhibitor. Due to its close structural similarities with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind, and as a result, enzyme action declines. Inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogens. Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on type of reaction they catalyze. Enzymes are divided into six classes, each with 413 subclasses and named accordingly by four-digit number. So there are six classes: oxidoreductases and dehydrogenases, transferases, hydrolases, lysases, isomerases, and ligases. Oxidoreductases, enzyme which catalyze oxidoreduction between two substrate S and S dash S reduced plus oxidized S gives S oxidized and S reduced. Transferases, enzyme catalyzes a transfer of a group G between other than hydrogen between a pair of substrate S and S. Hydrogelases, enzyme catalyze hydrolysis of ether peptide glycosidic C-C bond, C hydride, and PN bonds. Lysases, enzyme that catalyze removal of groups from substrate by mechanism that other than hydrolysis, leaving double bond. Isomerases include all enzymes catalyzing interconversion of optical geometry positional isomer. Ligases, enzymes catalyzing the linking together of two compounds, that is enzymes which catalyze joining of CO, CS, CN, PO bonds. Cofactors, enzyme are compound enzymes are composed of one or several peptide chains. Enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituents called cofactor are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzyme is called the apoenzyme. Three kinds of cofactor may be identified: prosthetic groups, coenzymes, and metallones. Prosthetic groups are organic compounds and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen. Heme is heme is prosthetic group, and it is a part of the active site of enzyme. Coenzymes are also organic compound. Coenzymes are also organic compounds, but their association with the apoenzyme is only transient, usually occurring during the course of catalysis. Furthermore, coenzymes serve as cofactor in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins, that is coenzyme nicotinamide adenine dioxide (NAD) and NADP contain the vitamin niacin. The number of enzymes required metal ions for their activity, which form coordination bonds with side chains at the active site and at the same time form one or more coordination bonds with the substrate. That is, zinc is a cofactor of the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. So, thanks for listening, and please subscribe my channel. Be curious.